benissimo, c'è la VGA qui. Okay, so welcome to the session of the afternoon. And now we have a session which is dedicated to how Italy can benefit from the Human Brain Project. And I mean, one of the purposes of the Human Brain Project is to reach the outer community, the neuroscience community, which is not within HBP, which is, I mean, the majority, of course, of the scientists is represented there. And so we invited Fiorenzo Conti, who is the president of the Italian Society for Neuroscience, which will talk about that and his idea and perspectives on what HBP should do to reach the outer community. So we are really looking forward to know from Fiorenzo what we should do to have the best benefit in Italy from HBP. And I also would like to show, we would like to show with Jim Miratto instead from Next Pisa, that in Italy there are similar initiatives called Flagship as well. And one of those is called Nanomax, and a big part of that project is related also to neuroscience. So let me call at this point Fiorenzo, please, and let's thank him to be here.
Vediamo se tutto funziona. No, l'abbiamo provato prima, ma funzionava tutto, quindi stiamo tranquilli. Dobbiamo esserci. Ok. Good morning and good afternoon, I would say. Let me first thank Francesco Pavone for inviting or for giving voice to the Italian Society for Neuroscience, and to warmly welcome you to our country, in particular to this city, and to remind you, for those who probably don't know it, that in Florence, Galileo Galilei, one of the founders of modern science, spent the last part of his life. Probably not the luckiest, but he lived here, and his grave is in the church of Santa Croce, which is a nice place to visit. I would start saying something about the Italian Society for Neuroscience, because I'm paid for that. And SINS, which is the acronym of Italian Society for Neuroscience, is a large society with more than 1,000 active members. And we have the know-how from ion channels to human diseases. And I think that in that environment, all people interested in studying brain, mind, will find the possibility of very positive collaboration. So our doors are totally open to people interested in whatever aspect touches the brain. Because we are convinced as society, and me personally, that the greater the mass and the most likely the possibility that we can get something important for our society. Society, not of neuroscience, society in general. And that is the major point that I would say as president. We are convinced, again, that contamination is the best way to stimulate creativity, and there are no doubts about that. And I repeat, we are totally open, and we would welcome whoever would be interested in collaborating with us. This is the answer to the question posed by Professor Pavone, but I'd like to say something more. I don't like very much these useless, often boring presidential speeches in which one say that his society is the best in the world. What we do, what we can do, and what we could do is written in our history that dates from almost 50 years now. And I think that most of you know at least some important Italian neuroscientists. It is not a case that most of the colleagues who got grants from HBP, who belong to this HBP club, are active members of the society. One of them has been a president very recently. He's sitting there, is Enrico Cherubini. And what I'd like now to address is something a little bit more complicated, less political, if you want, and more philosophical, if you want. And is to give you some of my reflections on how we do science now, and what are the risks and what are the benefits of doing science now compared to what we learned. What we learned is this. We grew up with the idea that research is basically an individual enterprise, or something that is done in a small group. And this kind of science, and we like to see the images of Cajal looking at the microscope, or Eccles working on his electrophysiological recordings, and so on and so forth. This science still lives. In the middle of the 20th century, around the 40s, something changing. And it changed abruptly. In the United States, they started the so-called Manhattan Project, which was a very wide, military-oriented scientific enterprise based on totally new concepts. The concept was huge amount of people, huge amount of money directed by politics. Just to summarize, okay? And it is not a case, at least in my opinion, that one of those who were involved with a big responsibility, this gentleman here, named Alvin Weinberg, here with J.F. Kennedy, 20 years later, published a book entitled Reflections of Big Science. It was not a case because Alvin Weinberg is the scientist who coined the term big science. And in this book, he expressed his experience very lucidly, very realistically. And what is even more remarkable is that several years before, in July 1961, Alvin Weinberg wrote a short essay in Science magazine entitled, as you see, Impact of Large-Scale Science on the United States. And it is a piece of literature, and really this should be a must for all young students in whatever branch of science you want. Everything we are dealing now, or we dealt with 2 years ago, or 10 or 15 years ago, was already clearly depicted in 1961. Let's start with a few questions Alvin Weinberg raised. The first, is big science ruining science? Is big science ruining us financially? Should we divert our money to other fields? We were in the 61, and as everybody knows, in those years, military spending was very, very high. And all the disciplines that were somehow tied to military were given a lot of money. And this reading is just fantastic. He says, I repeat, very, very lucidly, that yes, big science is probably ruining science, and he explains why. He says, one, because to have big science, you have to have big money, and to have big money, you have to convince the politicians, and to convince the politicians, you have to convince the people, so you have to get a lot of consensus. And thus, he concludes, you have to be somehow journalistic, which is fantastic. He says also that big science requires a huge amount of money, and that in his opinion, what scientists should spend are thoughts and not money. And he described how instead of buying a $1 million apparatus, you could probably build your own for much less. And third, of what Weinberg calls the diseases of big science is a huge administrative apparatus. And he says things very, very tough and rude. And he say here, unfortunately, science dominated by administrators is science understood by administrators, etc., etc. And he calls these the three diseases of big science. And he called them journalitis, moneitis, and administratitis. These are the three inflammatory diseases Weinberg devised. But he was a very intelligent man. And he was not only saying that something was not good, period. He also proposed how to bypass the problems. And he says here, we cannot do anything. Big science is here. What we have to do is we have to learn how to manage it, etc. The idea of big science moved from physics, as always, everything originates somehow from physics, and moved to biology and more recently to neuroscience, and we are here to talk about that. And the idea that big science exists and is useful is now accepted. This is an image taken from a very recent article from a physicist linking the concept of big science that is inevitable to him to big data, that is the next or the actual frontiers. And then big science moved to neuroscience and moved almost simultaneously here and in the United States with two projects whose general aim was similar, but that were very, very different in many aspects. The structure of the project was different, the management of the project was different, the way the project was conceived and realized was different. In the European project, most came top-down from politicians and down to researchers. In the American initiative, the original steps were in that direction, but were somehow stopped sufficiently early by neuroscientists who contributed with 1 year of long and public discussion to shape the project. I know very little about the American project, but I know, as all of us know, that our project has had problems. And these problems were not limited to our societies or to the community of neuroscientists. Everybody knew about it, and this went on General Magazine. This is taken from a 2015 paper in Scientific America. And we all know, and you all know the story, and I have not time enough to discuss. But what is important is not the detail. What is important here is the concepts that we can trace and the lesson we can learn from what happened. And here, you see, they ask themselves rhetorically, was it poor management or is something fundamentally wrong with big science? And we have seen it. There is something fundamentally wrong with big science. The problem that I see, and these are personal reflection, is that all the problems and the risks of big science had been clearly written in 1961. And had our politicians read this paper or understood what was going on, probably these problems could have been much, much reduced. There was clearly some planning that was not perfect, and there was some management or another aspect of management that was not perfect. I refer to transparency. And all these brought a crisis which has now been fixed, and this is, I hope, I took the very last slide, the final project, Hbp, with a new management, and we are all happy about this. It's clear that there are forces that tend to increase the number of subpanels. I'm sure this is an everyday experience for the big boss of the project. And in some way, it is normal to expect that because those are not all the aspects of neuroscience, and we all know that. But we also know that if we change too much this schema, we can alter the identity of the project, and we can make only a copy of the other initiative. So this idea, the original idea, was clear and was very personal. It had an identity. The problem that I can see now, and I'm finished, is that if we change too much these, we alter the identity of the project. The project has a great meaning, and all of us look forward to see results coming from this project. I think we should not change too much. Otherwise, we go into the camel paradox. As everybody knows, a camel, for many, is a horse drawn by a committee. And I'd like our project to maintain his identity. Thank you. And now, oh, sorry, the most important. Scusi, mi ridà la diapositiva. Okay, and now, enjoy the meeting, enjoy Florence, and spend thoughts and not only money in Florence. Thank you.
Thank you, Lorenzo, for the very nice and inspiring talk. I think we are really late, so let me maybe switch rapidly to Jimmy. By the way, Lorenzo will be here, so if anyone wants to ask him something, maybe after the session, Lorenzo will be
Vai.
Grazie.
Tu devi lavorare. Va bene.
Beato te. Sì, farò del mio meglio. Allora, partito? Ok. Thank you very much, Francesco, for having me here. I've been asked to tell you a little bit the story of this consortium, but of course Francesco organized it. It's called Nanomax, and so he should be the one actually giving this lecture. But in this talk I will give you a very, very brief outline of the project, and then I will dive a little bit more about the part that we are actually coordinating in Pisa, and I'll show you some results that hopefully might be interesting for a neurobiology audience like the one that I expect to have in front of me today. The name of the project is Nanomax. It's a flagship, but compared to the Human Brain Project, it's a rather small flagship, I would say. It's about 1 meter for every 100 meters of the Human Brain Project. But still, it was a very, very interesting thing that Francesco organized, because he organized a network of labs that comes from very, very different backgrounds, from physics, from nanofabrication, photonics, all the way to basic scientists that work in life science. And the idea was to put together life scientists with nanotechnologists in order to bring about let's say, well, the next generation, or at least another generation of sensing devices that could be used in a clinical setting. And the fundamental idea here is that paradigm of micro and nano fabrication and microfluidics can actually help providing different tools for detections of biomarkers. So there are several partners. You see them on the left. There are 7 main partners, and CNR participates with 7 different projects. And the project I'll be talking a little bit, which is the one I have the pleasure to coordinate, it's called NanoBrain. There are 33 CNR institutes involved. So we are talking about a rather large number of scientists that are working on this project. Just to give you an idea of the size of the consortium, the crop of 2015 has been of 125 publications. There are also several patents that have stemmed out from the collaborations. And we are about 144 investigators altogether, and 35% of them are actually under 40 years of age, which is surprisingly low age for Italy. I mean, we are all aging very, very much. It's sad to say, but that's exactly the way it is. So NanoBrain is a project, is actually a small project, involves only 3 institutes, which is my institute, the Nanoscience Institute, which is based in Pisa, the Institute of Neuroscience, and the Institute Officina dei Materiali, which is an institute very much devoted to nanotechnology. And the idea of NanoBrain is actually to study a very terrible pathology which is called glioblastoma. I would say, is arguably the most terrible brain tumor in existence. And we have 2 different actually set of studies, in vivo studies, where we are trying to develop novel actually models for basic study of the glioblastoma. And these models are actually important for the second platform of the grant, which is the ultrasensitive detection of the biomarkers. At this stage, we already have several devices, both in Padua and in Pisa. They're actually working up to a state-of-the-art sensitivity, and we are almost at the stage of testing them with samples coming both from the murine models and from patients. But I would like to give a more neurobiological, let's say, fingerprint to this study, and mentioning something that many, many years ago actually hit my head like a hammer, which is the fact that most of the pathways are really important for synaptic plasticity. My lab basically studies synaptic plasticity, at least in some of the many flavors in which you can study this. But several of the pathways that are important for synaptic plasticity actually are also important for tumor. So the idea we had in the lab is, okay, is there something that comes from the study of synaptic plasticity, from brain plasticity, that we can actually use and import it to give, let's say, to open a new window on the study of tumor, and specifically of brain tumor? What has in common? I mean, it's a very complicated question, and so you have an unreadable slide. I know it is unreadable, but it is unreadable on purposes. This is an outline of all the pathways really, really fundamental for cancer research, but all the pathways are coming out now, which are highlighted by these areas, are actually the pathways that we know are fundamental for synaptic plasticity. And you can see that there is really a massive overlap, especially here in the area of the MAP kinase pathway. Here we have PKA, and here we have all the molecular recognition, which is at the basis of the interaction of neuron with the extracellular matrix. And you see that all these elements, which are so important for synaptic plasticity, are actually also fundamental for cancer research. Then there are pathways not that important for synaptic plasticity, like the P53 pathway. I mean, they are basically quality control during the cell cycle, because neurons don't have a cell cycle. They're terminally differentiated, so obviously there are differences. And probably this important difference of neurons that do not differentiate further, that do not proliferate, is what has left all this pathway almost, devoid of function. So they start to do something else, which is synaptic plasticity. I know this is a very simplistic thought, but maybe that is something that is actually occurring in the brain. So one of the characteristics of glioblastoma that makes it so terrible is that it is very strongly invasive. And since the brain has a very diffuse function, all cases of glioblastoma, the diagnosis only occurs very, very late, basically, when is way too late to really have any successful treatment. As a matter of fact, of all the brain tumors, all the forms of glioblastoma have a median survival of less than 10 months. So that is as bad as it is. What are the questions that drive our project? we have no understanding of the early phases of the tumor, because basically we have no way, I mean, from the clinical point of view, of analyzing samples that comes from a tumor that just started. So we don't know what happens at the beginning of the proliferation. We don't know what happens at the beginning of the infiltration process that brings about the metastases, which are largely responsible for the terrible prognosis of the tumor. So we thought, okay, can we actually model that in murine models that then we can investigate by using two-photon imaging and all the tools of neurophysiology that we have? The answer is yes. There are different ways in which you can actually build up a tumor. And the example I will show you actually comes from a well-established model. Is a cell line. It's called GL261. It's a glioblastoma cell line which has been isolated in mouse. So this is, you can take these cells, you can engineer them in different ways, and then you can transplant in a mouse. You don't need to do this in naked mice because it is a syngenic transplant. And the dogma, at least what we know until now about this tumor, is that this is a very encapsulated tumor. You can see it from this image from Matteo Caleo's lab, still in Pisa, and is a member of Nanomax. It seems to be a very encapsulated tumor, and it grows as of course, a fatal prognosis for the mouse, but it doesn't seem to be very infiltrating. But I will actually show you that this is not true once you actually change the tools you use to look at it. So what we did is that we have taken these cell lines and we have engineered them in order to make them visible. We have this tumor now growing in very many different colors and with different genetic backgrounds. And you can take this cell and you can transplant them in the mouse. The tool we use, of course, is two-photon imaging. Here you can see a cranial window on the brain of a mouse. You can see the blood vessels. Here the bone has been exported. There is a cover slip. The chamber is sealed, so you can actually perform the imaging in several different days, and you can go back and look what happens inside the brain once you analyze it under the two-photon imaging. So this is what we did. This is a transplant. After 8 days from the transplant, the tumor is red. You can see there is also some green fluorescent underneath. This is actually very important, but I will tell you in a second what it is. Here you can see a little bit of the bone that surrounds the tumor. And after 8 days, you can see that the tumor at this stage is about 2 millimeters. The long axis is just under 2 millimeters, so it's still pretty small. But you can see that it's already very, very dense. And it has got a very interesting, and we were a bit surprised actually to see a very strong morphological differences from the central part of the tumor to the more peripheral parts of the tumor. And you can actually see this better, okay, in an image, okay, which is here. In this movie, you can actually see this is a three-dimensional trip in the brain of a mouse after the transplant. You can see it's a very, very small tumor. This is in vivo imaging. We comes from the surface of the brain and we travel inside it. And you can see that cells at the edge of the tumor actually have a very striking morphology because they look elongated, whereas in the central part of the tumor, they look more spherical. Keep in mind that this is a clone. I mean, it's a cell line. So all cells should really have the same genotype. And what we have here is actually that there are different interactions between the environment, which is the brain parenchyma, and the cells, which actually are doing something to the cells in order to change their morphology and polarization. So we see this in a magnification. You can see on the left, on the periphery of the tumor, the cells are very strongly elongated. And again, you see this green fluorescence. We'll get there in a second. Whereas in the center, they are almost look like a confluent culture. Not much seems to be happening. They are really very spherical, as it quantified in the images down here. So what are the green structure I'm showing you? We'll get there in a second, because we were very curious about something else. how these elongated cells are in terms of their structural plasticity. We know that in the brain there is a vast amount of structural plasticity, dendritic spine, both dendritic spines and synapses continuously change shape, but also astrocytes are affected by that in response of many different kind of stimuli. Well, also this cell have a very strong amount of plasticity. Actually, the spherical cells nearby the center, when they have the spherical morphology, you don't see much plasticity at all. You can see it very well here. But once the cell assumes this polarized phenotype, they're actually very plastic. You can see lots of lamellopodia sticking out, lots of blebbing, and in certain conditions, you can also see lots of phylopods that actually gets created. So, what is it that might be going on? And actually, you're getting some indication from this experiment here. So this is 1.5 hours of imaging in the brain. And you can see something interesting. We are very much at the edge. And you can see that the cells are all separated one from the other. So the density of the tumor here is very low. And you can see that most of the cells are actually parallel. You can see blebbing going on the edges of some cells, but you can also see phylopodia. And if you look carefully, you can actually see the phylopodia are moving a lot. So they are clearly exploring the environment. And again, you can see the green fluorescent underneath that actually seems to be providing a sort of vectorial field in which the cells are orientating. What is the green fluorescent? The green fluorescent is second harmonic fluorescence. This is something that is not very exploited in neuroscience, but is actually a very, very interesting physical phenomena. Certain kind of molecules, actually, once they are excited in the infrared, can actually provide a fluorescence. It's not fluorescence, but I'm using this word just to make a point. They actually release photons at a frequency which is exactly twice as much as the excitation. So here we are exciting 1040 nanometers, and we are getting this backscattered signal at exactly 520 nanometers. This signal comes from collagen. Basically, in the brain, the only molecule that can provide a signal is collagen. And you can see that the collagen fibers that are at the surface of the brain are actually providing a sort of scaffolding that control the polarization of cancer cells. We are very excited, obviously, about these results. I mean, there is plenty of literature in oncology research showing the interactions of a scaffold, of extracellular scaffold for tumor cells. But here we actually can exploit dynamic two-photon imaging to study the time-resolved and space-resolved interaction between collagen and brain cell. So here we have a clear indication that the scaffold, the extracellular scaffold, controls the polarization of the tumor cell. But the second question is that also controlling the infiltration of the cells? I mean, it's not the same thing to orient the cells and to orient their motions. And this is something you can see in this experiment. This is a chronic imaging experiment in which the tumor has been imaged for several days. I'm just showing 2 days here, right after the transplant. In blue, in cyan, we have the second harmonic signal originating from the collagen filaments, and in red you have the cancer cells. And this is 8 days after the transplant, and you can see that most cells are very tightly oriented in the direction of the collagen scaffold. And if you look at the day after, you can actually perceive movement, and this movement is due to the fact that the cells are actually migrating, and they're actually migrating exactly in the direction of the scaffold. So, for example, if you look at this cell here actually will move, and you can see that it keeps moving along the filaments. And this is true here. For example, this cell here creates a very long phylopodia that moves to the left and trigger the migration of the cells towards the left. This image here, actually, you can see overimposed the imaging from 2 different days, and you can really work out the fact that these cells are actually moving along a sort of vectorial field, which is actually dictated by the structure of the extracellular collagen. Why we are very excited? Well, because at this point we can study the pharmacology of this interaction, and we can try to exploit it in order to control and influence the infiltrative properties of this tumor. What are the future perspectives of this? Well, our idea is to do a sort of fingerprinting of our glioblastoma models, both exploiting the spatial and temporal resolution that we can have because we are an in vivo model where we can actually recognize the tumor cells because they express a fluorescent marker. The second thing that I don't have time to get into details, is that we are actually developing a on-demand model of glioblastoma, which does not require any transplant at all, but it only requires viral vector to cause a specific mutation in very small subset of cells, and together with fluorescent markers that actually describe exactly the genotype of the cells, so that by using, again, two-photon imaging, we can actually study the temporal evolution of these processes. The final point I want to make is that once you have tumor cells, and this you can extend to any kind of brain disease, in which you can insert your mutation in a subset of cells, and then you can distinguish this mutated subset of cell with a specific fluorescent marker. What you can do the proteomic and the genomic fingerprinting of these cells. Here we are exploiting a collaboration with Liam McDonald that is in Pisa, and he's using laser capture to capture the cells that express the mutated genotype. Since we can recognize them by the fluorescence, and right now we are screening a number of proteic results that are coming just by extracting different portions of the tumor as we are guided by our fluorescent marker. Thank you very much. I hope this was interesting, and have a nice day in Florence.
Thank you, Jimmy. I would propose for this session, but also for the next session, maybe to ask questions to the speakers during the coffee break, because we are really late, and I mean otherwise we have a problem with the program. So thank you for the nice talk, Jimmy, and let me open another session, which is HBP in Italy. So it's a great honor for me to open these sessions, and also can I call Egidio? Can you please come here with me? And let me say one thing. I mean, I'm proud to host you here and to host this HBP Italy. But let me say that at the beginning we had a competition, and in Florence and Pavia, they were competing together. So Florence got this honor, but I would like to say that Pavia is also a great place, great science, and I mean maybe next time we will have this opportunity. So I like to share with Egidio this kind of session, and HBP Italy is maybe something that we think it's very important for us in order to coordinate each other. Do you want to say something, Egidio?
Yeah, thank you, Francesco, for the very nice and kind introduction. We had two towns that were relevant for HBP. It's an honor to be here in Florence. Hopefully we will have some very nice event in Pavia in the future. I think what is important now is to make give a picture and make a framework for HBP in Italy. I think now we are many relevant and well-working groups, and I hope that this session of today, these two sessions of today, will help all of us to understand exactly what is the target that we can take all together. So thank you, Francesco, for the huge organization. I understand, I mean, in a way, when he won the competition for the town, I was relieved. Because, I mean, organizing, I had an idea of what kind of adventure I was trying to go through. And when he won the competition, I mean, I was in a way, thank you, Francesco, for doing all the organization. Thank you very much.
Okay, thank you, Egidio. So let's start. Okay, so may I suggest to the speakers to be very precise. We have 10 minutes each one, and to be really, really precise in time. So the first speakers will be Marcello Massimini, and he will talk about consciousness and brain complexity from the macro to the micro scale.
All right, thank you, Francesco, and the organizers. I will ask you to follow me for 10 minutes sharp in a quick journey in the brain, searching for, exploring the relationship between consciousness and complexity in cortical circuits at different scales, at different levels. So, we are involved in two tasks. We just entered the Human Brain Project. I won't go into the technical details of the task. I just want to give you the flavor of what we're doing. These are two tasks that are diverse, but also synergistic. But what's important is the way we look at the brain, which is the tools that we use. That is, we just do not observe the brain. We also perturb it. So we basically stimulate the cerebral cortex and record the reaction of the rest of the brain, which is a peculiar approach, which is causal rather than observational. We do these perturbations, as you will see, at different levels: macro, mesoscale, microscale, and we do it non-invasively and invasively in different ways. So what's the advantage of doing this? And we can record the immediate response where we perturb. This is a measure of excitability, local reactivity. We can measure how it propagates. It is a measure of connectivity, true connectivity, causal connectivity, which is important. We can also measure the complexity of this pattern of causal connectivity. And complexity is important because it is theoretically important for consciousness, at least, according to the up-to-date theories of consciousness. And we'll see whether this is important also in practice. This is one of our goals. So we have these kind of measures, and what is relevant for consciousness also is that we are performing these measures since we're perturbing and recording directly from the cerebral cortex, independently on input and output. And this is practically important for patients who sometimes are disconnected. They don't perceive from the environment, they cannot move because they're paralyzed. So this is just a practicality, but it is crucial. So, two tasks. In one task, we explore the relationship, how intrinsic neuronal properties affect overall neuronal brain complexity. And in the other task, we move to patients and we look at how cortical lesions affect complexity. Give you an example of what kind of measure we perform. Macro scale. So we need to perturb the brain, and we do this by using transcranial magnetic stimulation, brief, strong magnetic pulse, which activates cortical neurons. And then we can navigate the stimulation, we can know how strong, where we're stimulating, we can be reproducible, have a good control on the way we stimulate without opening the skull. And at the same time, we can record the overall response of the brain with a good spatiotemporal resolution by putting these sensors on the head. This is basically electroencephalography combined with transcranial magnetic stimulation. So this is the tool. And we started using the tool in 2005, almost more than 10 years ago, actually, because we were curious we had a question in mind. So we asked, why consciousness fades when you fall asleep into non-REM sleep? It's a striking fact. We always forget about it, but at some point, we don't exist anymore. When we sleep heavily, we don't exist, and the entire universe does not exist. And there's no good answer in terms of neuronal activity, because neurons are active during sleep, they are reactive, so that was a mystery, and that's why we started, using this perturbation. Let's knock on the brain and see what changes in the brain, the way the brain connects and communicates within. So this is an example of what happens in an awake, conscious brain. You stimulate, you record these traces, and then you look at the spatiotemporal dynamics, and you see that you have a complex spread of activity. It's a complex network which gets activated for 300 milliseconds. It's not a long time for us, but it's a long time for the brain. So this is a complex spatiotemporal dynamic. Then, when consciousness fades, non-REM sleep, stage 3, you don't exist anymore, this is what happens. The brain is active and reactive, but look at the spatiotemporal dynamics. So this is a striking difference. As striking as the difference you have in personal experience. You don't exist anymore. The response is local and simple. So there's really a big change in the brain. Although the structure is there, something has changed functionally. and it's a big change. Now, this is eyeball complexity, just a description. We try to quantify this complexity, and the way we do it is that we record the spatial-temporal dynamic, we zip it, as you would do with a file on your computer, and we measure the complexity by compressibility. The more compressible, the less complex, and the less compressible, the more complex. And then we test this measure, over the years in many different conditions: wakefulness, non-REM sleep, midazolam, propofol, xenon, different aesthetics, you name it, ketamine, anesthesia, during which you are completely unresponsive, isolated, you look like in a coma, but you're conscious because you dream a lot, and complexity is high. During dreaming, complexity is high. During wakefulness, complexity is high. And during states where you're knocked unconscious, propofol, xenon, and sleep, and so on, complexity is low. So it's a good matrix, it works well. And, after all, we've been trying it in, many subjects, different conditions, including brain injury patients, and we can say that this theory-driven matrix of complexity is a good, reliable index. Now, this is the background. Within the project, we now are ready to test, after the validation, to test the measure in patients. And here the challenge is, it's not only scientific, it's also a clinical challenge, because oftentimes patients emerging from coma are unresponsive, but they might be conscious. But we don't know, because they cannot move and they don't understand commands. So, measuring complexity in these patients, extreme case of cortical islands, because now we save people from coma and we save islands of the cortex who might be disconnected from the external environment, it is crucial ethically, scientifically, and clinically to detect consciousness in these patients who are disconnected. So this is, the first actually result of our activity within the HBP with this paper, where we are able to stratify unresponsive patients based on their direct response to a perturbation, cortical perturbation. So you see there are patients where the response is flat, cortex is gone, not much to do here. Patients in whom the response is a sleep-like, low complexity response. They are a mystery to me. And then there are patients in whom we see complex response, just as complex as the one we observe in awake patients or subject waking up from a dream and telling you that they were conscious. So we need to assume that these patients and these cortical islands are conscious, and then we need to find ways for this patient to communicate. This is clinically, obviously, and ethically relevant. But I want to focus on this patient, the low complexity patient. Why is that? This is a sleep-like response after brain injury, and these patients are stuck in this state. An example, this is a patient where the entire right hemisphere is preserved, high metabolism, as detected with the PET scan, but a low complexity. And he's vegetative. He's been vegetative for 5 years. So the question is, can we get it back to a normal level of complexity? Can we restore, push consciousness, and bring it back to the surface? Is there anything we can do? After all, sleep is reversible, and this is a sleep-like response. So, we need to think about what's behind this, and we have an hypothesis that this kind of wave reflects the fact the neuron at some point becomes silent, and they forget about the input they receive, so complex pattern cannot arise in simple terms. So basically, we need to demonstrate first that this is a silent state in the brain, linking neuronal activity to overall complexity, and finally to consciousness. So this is our task, after all. So, we go one step deeper, and we go inside the brain, taking advantage of pre-surgical evaluation of epilepsy patients, where we can perform stimulation and recordings. This is done clinically, routine clinical exam. We just take the data, perturbations and recordings in different states, wakefulness and sleep. And we see, if we go inside, trust me, during sleep, this blue thing is a neuronal silence. So you stimulate the brain, the neurons are becoming active, and then they shut down, and they forget, because they become silent. So we need to link this to complexity and test whether this is actually relevant in killing complexity. We can do this, and this is really new, by adding on top of the head of these patients a 256-channel EEG system, which is also useful for clinical purposes. This is the main purpose, but we take advantage of that, and we can link this mesoscale closer to the neurons, to the macroscale. So here we can observe the pattern of, responsive to the perturbation, and we can measure the complexity of the pattern, but then we can also relate this global scale dynamic to neuronal events recorded deep in the brain, closer to the neurons. And what we see, these are preliminary results, is that actually this downstate, which is a neuronal silence, which is very well known, the mechanism we know, is a killer for complexity. Complexity stops growing after this blue area. So now, of course, you know, once you have a phenomenon, you want to see whether you can actually try to revert, and you know, the idea is that you have to revert this downstate and restore complexity. We tried this in the slice in collaboration with Mavi Sanchez-Vivets, again within SP3. So this is a slice who's asleep, you have this downstate, you can manipulate pharmacologically the slice and you wake it up just by closing potassium channels, for instance, and you restore complexity. So overall, our goal is to link consciousness to complexity through the mesoscale to the microscale, design new interventions at the microscale, and then go back and feed it back to the patients. And of course, simulations here play a key role, and I think, you know, interacting with the platform in HBP will be a great opportunity for us, and indeed we have many collaborations, and we are going to have many collaborations with different tasks within HBP. Modeling will be important, you know, to understand why consciousness and complexity collapse and recover in the thalamocortical system, which is a scientific mystery, but I think most important, you know, an understanding and seeing whether we can revert complexity in the brain after a lesion. I think this is a, you know, major thing, because after all, complexity collapses after any cortical lesion, including stroke, and recovery of function is linked probably to recovery of complexity. This is a dream, but it fits with the HBP, because I think also this project is a dream overall. Thank you, and thank to all the collaborators.
Thank you, Marcello, for a very nice talk. So Marcello Massimini from the University of Milano. And then we move from Milano to Pavia with Egidio D'Angelo talking about HBP neurons and microcircuits modeling.
Trovo la mia soluzione che vengo da solo, guarda un po' se da solo. Cosa deve succedere? Okay, yes. We got it. Good. So the activity that I will present you inside the Human Brain Project is devoted to the investigation and modeling of neurons and microcircuits. Why we are addressing this issue? Before going to that, I mean, because many of you already know what we're doing, I mean, I will start by telling you where we are placed into the Human Brain Project. We are working into the SP6. The models are constructed and supported by SP6. And we coordinate this modeling activity through several sub-projects, including SP1 for the for biology. In SP2 and 3, you have seen what Marcello has just shown. We have some ideas to collaborate on this in order to measure complexity in the circuits. That's one of the main issues to understand how the circuits process information. Then we are working together with SP5 in order to process the data, SP7 for supercomputing, and the new with SP10 for robotics, and with SP8 for medical informatics. This is a new adventure that we are starting right now. The action of our group is extending over the territory. We have several collaborations in Italy, but in particular, I wish to illustrate what we're doing in Lombardy. We had a discussion just an hour ago at lunch on how HBP could integrate into the Italian reality for research. One way is to leverage on the regions. The regions may be helpful, can be helpful. In this case, the region helped us to coordinate the action and the research in Lombardy in different centers, including the University of Milano, where Marcello is now, University of Milano Bicocca, the second University of Milano, with the Politecnico di Milano, where we work on robotics. With the Ospedale Niguarda, that's a new opportunity that will appear soon for medical informatics. and with the IRCCS Institute for Neurological Research, Mondino in Pavia, again for approaching integrative neurophysiology and neurology. So fundamentally, we are trying to integrate and extend the activity outside the university toward other institutions. What are we doing? But we start from basic principles. I don't tell the story of Human Brain Project. It's a wonderful dream, it's a wonderful vision, and I wish just to tell you what we're doing inside this big architecture, this logical architecture of science. We work on the cerebellum. The cerebellum is the second main cortex of the brain. Is less known and less investigated than the cerebral cortex, but it is very important. Recently, in our group, but I would say in many groups, there have been evidence that the cerebellum is involved in cognition as much as it is involved in motor control. So in order to understand how areas of the brain communicate one with each other and cooperate in generating integrative functions, we needed to understand not only the cerebral cortex, but also other areas which are fundamental, the basal ganglia, the hippocampus, and the cerebellum. So we are on the cerebellum. And this investigation has been recognized as strategic for HBP in the SGA1, so in the phase in which we are now. Investigating the cerebellum for a neuroscientist means investigating the molecules, the neurons, the microcircuits, the local circuits on the mesoscale, and then the integration of the structure on the macro scale. What happens in the lab? We are taking advantage now of our integration into SP1, and we're pushing on the biological part much more than in the last 2 years. We work on the neurons, we can record activity from the neurons, we can do precise reconstruction of neurons. There is an interesting collaboration with Javier in SP1. Then we work on the field of two-photon microscopy, high-resolution techniques for understanding the activity of brain circuits. That's a critical issue. We can't do with the current techniques all what we would like to do because the techniques are limited. And there is really a need to develop advanced techniques, and Francesco is pioneering in this field. These techniques allow to investigate more than one neuron at the same time. That's the nightmare of the microcircuit, because we all think that we are able to investigate the microcircuit. We are not, because we do not have the techniques, the technologies, to measure activities simultaneously at the resolution of spikes in all the neurons that we need to understand and to measure. But this is an effort. We're implementing several of these techniques, including optogenetics, multi-electrode array, high density, 4,000 electrodes in microchips, and two-photon microscopy designed in a way that we can measure activity from many neurons simultaneously. So this is the core of the laboratory. And the laboratory feeds information into what? Into microcircuit modeling. And microcircuit modeling is in SP6. So in microcircuit modeling, we use information coming from biology, and this information is used to reconstruct models of neurons, synapses, and then by propagation, we make the microcircuit. And the microcircuit is working pretty well because already at this moment, is capable of reproducing most of the properties observed with the experiments. For example, spatio-temporal redistribution of signals. You know that the cerebellum is one of the structures of the brain in which the spatio-temporal reconversion is hypothesized to be critical for converting space and time in the brain. There is something like a relativistic reminiscence here. And this reconversion occurs strongly in the cerebellum. So the basis for this reconversion are here. And we have to look for their correlates in neurophysiological terms. It's a bit like looking for the correlates of consciousness, as Marcello said just before. So we have to find the correlates of a fundamental property of the brain in physical terms. So the microcircuit is beginning to work nicely. There was a great effort in the last months to reconstruct it in the brain builder. And now this microcircuit is ready to work, and I hope that in the future we will be able to propagate the microcircuit to the meso and macro scale, reconnecting the cerebellum to the rest of the brain. This is one of the missions of human brain. We're doing a piece of this mission. And this work is correlated with that of many other groups, mostly in Lausanne and in Geneva now. The process of making models, you all know how this process is. We're following this strategy since many years. And what is most important is that by having a laboratory that integrates biology with modeling, we can go from biology to modeling through all the steps, contribute to the development of tools. At the same time, when we have the model working, then with this model we can make predictions, and we go back to biology. So we close the loop. And I think this is one of the most interesting adventures that we are facing now. I make you an example. This is an example in which we have a model of a neuron. This is a model of the Purkinje cell. It's one of the most complex neurons of the brain. I expected it was working. You should see the spikes coming out and the simulation be running, but at the moment it is, it's jumping a bit. It's more or less stuck. And this modeling is implemented in core neuron, and we are contributing also to develop tools for these core neuron facilities. What happens then is these models can be used to interpret the biological data, and this is one of the most interesting happenings in the lab in the last years. We had previous measurements of the mechanisms of action potential generation in the axons. In the axon of neurons that are the granule cells of the cerebellum, and we had predictions on how this mechanism could work. In principle, every scientist starts from what is known in theory. The theory says that Hodgkin and Huxley resolved the problem of spike generation in the giant squid axon. And this theory holds, and all the world is working on this theory. The problem is that when you put this theory into a neuron, you have to take into account that the neuron is made of parts that are not perfectly identical. You have a soma, you have the axonal initial segment, you have the axon, and when you put all of this together, the theory is not enough because you don't get the action potential in the proper manner. So we discovered it by using models. Because with what we know on the sodium channels and on the membrane resistance and on the structure of the neuron, when you do fundamental biophysics on this neuron and you put it in a model, it doesn't give out the spikes that they should be. So we decided to reinvestigate the issue, to develop new techniques, for example, voltage-sensitive dye imaging in single neurons. And what it came out is that the spikes do not behave as they're expected to do. We have done new models on this expectation. We have analyzed the biological mechanisms of this, let's say, specific feature of mammalian neurons in generating spikes. And what turns out is that now there is a new model done in the Hbp framework that reinterprets the mechanism of action potential generation with substantial changes. The axon is not leaky. Hodgkin and Huxley said it is leaky. It is not leaky, basically. The sodium channels should be the same in the soma and in the axon. They're not. They're different. So there are several changes, evolutionary changes that sum up and converge in making our neurons unique. Another nice adventure is that of reconstructing the network. This is a brain builder reconstruction that was achieved in the last weeks, I have to say, in which we are trying to re-implement all the knowledge on the cerebellum, which dates back to Eccles and very remarkable people in the last century, and has been taken as it is for years. Now this field is under revolution. It's all changing. So we need something flexible, able to incorporate the new connections. If you take a book on the cerebellum or a review, we know everything of the cerebellum. It is false. It's not true. We do not know everything. Every year there are papers coming out of a very important journal demonstrating that the cerebellum field is exploding. So we have to reinterpret also with the models. And this is the way to do that. I mean, we have a model constructed bottom-up in which we can introduce the variants and the novelties and rebuild our conceptions, our understanding of the structure. And this is an example of how it works. This is a model that is running on one of our supercomputers. I think this is the Lugano supercomputer for the moment. And this model is showing the activity of 400,000 neurons in the cerebellum simultaneously. And thanks to the graphics, it's incredibly, it generates incredible images. This is the last step. That's the last step that is being implemented. I'm going to finish. This step is the propagation of models into robots. And this is a robot that is producing spikes. It's a spiking robot. And the spiking robot is simulating a simple behavior. I don't tell you all the story, but basically this robot is implemented with neurons, with plasticity rules and nothing else. It doesn't know anything at the beginning. And it learns to behave and to interact with the environment in an appropriate manner. So which means that we are, in a way, trying to reconnect biology with engineering. And in a way, this works. That will require further development. One of the last attempts is that to put this sort of microcircuit in NEST. NEST is the platform for reconstructing large-scale network that is being used in HBP, and to put it in the virtual mouse model. It's a mouse with a brain that should behave like a mouse, but is a video game. And in this video game, there are not physical laws that govern the movement or the shape of the mouse. There are neurons, and these neurons control what the mouse will be able to do in the video game. So this is one of the last challenges. The very last is this, and it's the connection between all what I told you and medical informatics, because one of the targets of HBP is to reconnect and to project into into medicine. And on this side, we're interacting with SP8, and we are elaborating a system for analyzing the data on a big scale, big data, with machine learning algorithms in order to understand better how pathologies are generated based on biology. Again, this is the fundamental principle. Biology is the source and the root of everything in this case. I thank everybody for patience. There's the model collab. The cerebellum will involve many people inside and outside human brain. And this is also an initiative that is growing at the moment. Thank you.
Okay, thank you, Egidio. We leave the questions at the end of the session. And now we have from the Scuola Normale Superiore Antonino Cattaneo. May I ask the speakers please to stay in 10 minutes each one.
So thank you, ladies and gentlemen. What I'll be talking to you today is going from top levels of brain function to down to the very molecular level. And I'll be talking to you about a technology platform that we've been working on. Come si va avanti? Okay, sorry. I'll be talking to you about a technology platform that we've been developing, and this concerns antibodies. Antibodies are essential reagents for neurosciences and for all fields of biology, actually. They are the best molecular handle that we have on recognizing chemically with high specificity and selectivity biological structures. And everybody who has used antibodies in the lab knows that the low quality of many antibodies is a well-recognized bottleneck, and people have calculated the economic loss, the time loss, and how this is a real scientific problem. Second, genomic and proteomic studies generate large numbers of protein targets, and this is a protein expression bottleneck, and I will explain to you in a while, because you need proteins to generate antibodies. So you need to streamline the generation of large numbers of good quality antibodies. And most important, many interesting antigens are conformational and post-translation modifications of a protein target. So this means that there is a big importance in focusing on technologies to facilitate the derivation of good quality antibodies. Classical methods to obtain antibodies are you need to have a protein to immunize an animal, or with modern techniques, phage display, ribosomal display, you need to have a protein, and then you derive the antibody. Because it is difficult to handle large quantities of proteins in many cases, most antibodies are derived against peptide antibodies. peptide antigens. We have developed over the years a selection that is called intracellular antibody capture technology that has that within the Human Brain Project we are developing as a proof of concept to validate the technology as an important platform for the needs of the Human Brain Project. And this technology allows in vivo production of the native target protein directly from cDNA sequences, so from gene sequences to DNA to antibodies. And this technology is based on the yeast-to hybrid system. You have your target antigen that is expressed in a yeast cell, and you have, most important, the library of antibodies that instead of being expressed in the immune system of an animal or of phage, is expressed in yeast. And so you have a synthetic immune system in vitro, which is made by yeast cells, and then you transform these cells with your antigen. The cell with the cDNA sequence for the antigen, the cells makes the protein for you, and then the system is organized in such a way that when there is a positive interaction between an antibody in the library and the target antigen, an antibody is selected. This technology has been validated in many different, several years by many labs around the world, has many advantages. One advantage is that you can use the derived antibodies both as classical antibodies, except that they come in formats that you can engineer for useful formats for mapping and imaging and sensing and other technologies. So you can do everything that you can do with normal antibodies, except that you can do it in more sophisticated ways. But most importantly, since you derive also the genes that encode for that antibody, you can express then these genes inside cells and express them, as we say, as intracellular antibodies. We have pioneered over the years the use of intracellular antibodies, genes that code for specific antibodies that targeted to different subcellular compartments to make what we call a phenotypic knockout. There are many things that we can do in functional studies with nucleic acid-based interference technologies, but there are many things that you cannot do in principle with gene-based approaches, such as targeting conformational epitopes or targeting post-translational modifications. So the technology platform that we are working on, that we have developed, allows both to use antibodies as classical antibodies, but with superior properties I will convince you in a while, but also as intrabodies to do functional interference inside cells and interfere with specific processes. In one task of the Human Brain Project, we are exploiting the technology to isolate small antibody domains for next generation brain imaging and mapping. This task has just started in the new phase, and so I'm just showing you an example of what we can do. Of course, this technology allows for the isolation of small antibody domains that you can directly chemically label, so you can do mapping studies, avoiding the indirect immunofluorescence studies. They are smaller, they are more soluble, they are less toxic, and they have a large number of advantages that I cannot have time to discuss that promises these to be superior tools. And as an example, we have derived antibodies against a difficult target antigen, that is the oligomers of the amyloid peptide of Alzheimer's disease. And these antibodies have properties that make them very, very unique reagents, because they are both conformational antibodies and they are also sequence specific, so they recognize specifically a certain conformation of the amyloid peptide in the pathological form. And with these antibodies, we can, antibody domains with these nanobodies, we can map Alzheimer's tissue in ways that in the literature had been not possible so far. We can recognize different localization and different cell specificity and different forms of the oligomers with respect to the different stages of the Alzheimer's brain. But the same antibodies can then, if we take the genes for these antibodies, we can then express them inside cells that make the oligomer, and for instance, start asking a question: where do the Alzheimer's oligomers form inside the cell? unless you have a molecular handle on this, a precise molecular handle that distinguishes the monomeric peptide from the oligomeric peptide, you cannot simply address this question. And we, Giovanni Meli and collaborators at Ebri and at Scuola Normale, have demonstrated that with this tool, we can directly demonstrate that in the cell, oligomers are first formed in the endoplasmic reticulum, showing that you can interfere and block the formation of these oligomers. In another task of this, where we are involved with Ebri, Scuola Normale, and Michele Migliore at CNR, we go from derivation of antibodies against the proteins of the neuroligin and neurexin families, synaptic proteins, use these antibodies to interfere with synaptic functions inside the neurons, and then feed this data into a computational model of the synapse. And Enrico Cherubini will talk about this work initially done with gefferin, that is another synaptic protein, and this shows a pipeline of how antibodies generated from this technology platform can then be used for functional interference inside the synapses, going on to electrophysiological recording, and then going on to computational modeling. And this is a summary of some of the antibodies that have been already generated inside the project. But to finish my short presentation, I want to add a last development of the technology that we have made. This tackles the problem that I've called the proteome complexity. We all know that the proteome is much, much more complex than the genome, because in face of a few 100,000 transcripts or so-called, or so, the proteomic complexity is much larger. And this is because you have conformations, you have post-translational modifications, and you have variations in proteins, chemical variations that occur after the protein is synthesized and encoded by the gene. So this can be called the dark matter of biology. And if you want to study synaptic function, neuronal function, you need to be able to tackle these modifications from the point of view in ways that you cannot do with the functional interference methods that are based on nucleic acids. So we all know that it is with nucleic acids you can, in a protein network, you can hit the nose of a network, but you can, because you will interfere with all the links that the protein makes. But whereas post-translational modifications hit the links between a node and another. So the question is, can we develop a post-translational intracellular silencing antibody platform? Can we selectively interfere with a certain post-translational modification of a protein without interfering with the unmodified version of that same protein? And this is not possible by current methods. And briefly, I'll conclude what we have done. We have implemented the intracellular antibody capture technology to specifically target the post-translationally modified protein. And in work that is currently under revision in a journal, Michele Chirichella in the lab has demonstrated that he can use tethered catalysis. So he can introduce in the cell the modification that is desired, for instance, acetylation of proteins, and you can use this to select antibodies. And these anti-PTM antibodies have been then used to interfere in a cell specifically with the post-translationally modified protein. So to conclude, this final work extends to post-translational modification, our platform, and what we hope to demonstrate in our contribution to the Human Brain Project is that technologies such as the one that I have described to you will be important to contribute to the molecular dissection of synaptic and neuronal intracellular circuits and mapping and imaging at different scales. Thank you.
So thank you, Antonino, for the very nice talk. And now Cecilia Laschi from the Scuola Superiore Sant'Anna will talk about Neuroscience and Robotics, the HBP Neurorobotics platform.
Grazie, ma sarò brevissima, vedrai che sono più... So thanks for the introduction. I'm happy to have the opportunity to talk here at this open day for the Human Brain Project. Okay. So I'm from the Biorobotics Institute of the Scuola Superiore Sant'Anna in Pisa. So as you understand from the name, we are roboticists. So our institute has been doing humanoid robots, among many others, for several years. And it is this group on humanoid robotics which is contributing now to the Human Brain Project. So there is a lot of expectations about robots today. As you know, they are expected to help us in many everyday activities, but I mean, they are not still in our houses for the moment. But what they are doing well for us is to be very good tools for research in neuroscience, in fact. They have demonstrated already this. So there is a lot of research about humanoid robots and neuroscience, and this is what we do in our lab. And as you see, what we do is to try to get some of the brain models that are studied in neuroscience and implement them on our robots. Of course, as roboticists, we like to do that because we want to see our robots performing better in some behaviors, in some sensory motor coordination activities, in motor control. But at the same time, we are also giving a tool to neuroscientists to validate their models, to see their models in action working on an artificial being. And these are some of the topics where we use this approach. So neurorobotics is sometimes used with this meaning, the use of robots. So you build a physical robot, a physical body for testing brain models. And the neurorobotics platform of the Human Brain Project has exactly this meaning. So we want to build as complex body as the complex brain models that we are developing in the project. You see that the neurorobotics platform of HBP, which is sub-project 10, by the way, is a simulated environment like this one, where you can set up your own experiment. So it's for scientists, for neuroscientists. They can set up their experiment, and they can set up the environment, the robot, or even animal models, as you will see in a while. And of course, the brain models that have been developed throughout the project, and see them in action. So there are some robots already identified to be included into the neurorobotics platform. This one is also visible downstairs in the exhibition area. This is another example of a robot that is being included in the neurorobotics platform. And this is a view of how it looks like if you want to access it. It is an open platform, so you can just access it through the internet. Of course, you have to log in with some account, but then you can see several things. Now what? So, for example, there are tutorials showing how to use it, and there are some experiments already prepared. But of course, the interesting part is that you can set up your own experiment. As I said, including the environment, the robot or the animal model you want, and the brain model. Okay, this is one of the experiments already tested in the platform. I didn't, okay. So it's a basic robotics experiment. It's based on the Valentino Breitenberg hypothesis. So this robot is just looking at the looking for the red color, as you see. And in this other experiment, instead, this is a test of this mouse model. Which is very interesting because you can see very tiny deformations of the surface of the animal. In this case, the movement is only the movement of the head again towards the red color. Just to demonstrate the mouse model here. While these other experiments include a robot that you probably have seen somewhere, the Hbp platform. It's a platform for neuroscience, in fact, and it's a physical robot that is integrated in the platform in this case, and this is a tracking target. So it is tracking the visual target that you see on the blue screen. Interestingly, this experiment can be also done with the physical robots that eventually will be integrated in the neurorobotics platform. Okay, so just to conclude, I wanted to give you this very short view of the neurorobotics platform. I hope you got the main point. This is an open platform, and it's for neuroscientists following this approach of using robots to test and validate brain models. It's an open platform, as I said, so it's available on the internet, and there are already a set of robots that will be, of course, improved. And there are several visualization modes, so not only on the desktop, which is probably the most common, but also on mobile devices up to real caves where the brain models can be visualized in action. So thank you for listening.
Okay, thank you, Cecilia. And the last but not less important talk is from Giovanni Erbacci from Cineca about Cineca and the HBP high performance analytics and computing platforms.
Okay, so the Cineca platform. We have seen so far a lot of interesting science, and science often needs simulation and need computer. And in my talk, I will present the involvement of Cineca in the HPC platform in human brain. And for this, I must first introduce a little the system that we provide in Cineca for the supercomputer. Cineca is a consortium of 70 and more universities, is the National Supercomputing Center in Italy, with an HPC environment equipped with cutting-edge technology and provides services to the scientific community. Furthermore, Cineca is also the HPC infrastructure integrated in the European HPC ecosystem and is one of the 5 hosting members in PRES, the European HPC infrastructure. So we have a huge experience in supercomputing. Since 1969, when Cineca was founded, we moved to the system that were supercomputer at that time, till the new system called Marconi-Lenovo that has been installed in the first partition in June, and the second one is in the installation phase. We move from a power of 10 to 6 floating point operation per second, so from 1 million of operation per second to 10 to the 16 floating point operation per second in this time frame. So we have an increasement, a factor of 10 to the 10 in the power of the system in this period. So just to introduce the first system, the Tier 0 system that we provide in the context of human brain project, I said Marconi, is based in 3 different phases. Here you can see the number of core, And when everything will be completed, we have more than 350,000 core. It means that 350,000 laptop or something like that, that can be seen as a single computer integrated in a network to do huge simulation. All those systems is provided free of charge for the scientific activity for huge project and of interest for human brain. Furthermore, we have other systems like Galileo for production of medium scalability application, and it means tier 1, and Pico for it's a system for big data and data analytics application and support at the Italian research activity. So, given that, we move to the support and our involvement in human brain. Cineca is involved in the SP7 platform for the high performance analytics and computing platform. Is one of the 4 sites with Jülich, BSc in Barcelona and CSCS in Lugano to integrate a system of different supercomputing to support the human brain scientist in order to do the huge simulation. So we operate and we support the computer and the data system infrastructure that is needed for the scientist in human brain project. We give support and maintenance also to the middleware that is needed, and we build on the existing capability of the different sites, and we integrate everything in order to provide a best support, the best integration to the sciences. There are specific calls for access to the system, are based on the press call. I remember here that just yesterday opened a press call for support activity, and now there are 4 of the 5 hosting members in press are involved in human brain, and so it's a huge possibility to get access to this call in order to get the resources to run simulation in human brain. Here is another view of the 4 centers that integrate the high-performance analytic and computing platform, and those are interconnected with a specific network that is the PRESS at 10 gigabit network. And there are also other centers that can be connected in order to provide data to access to the simulation activity, to move, to do visualization and so on. This is how we provide the activity for the high-performance analytic and computing platform. Furthermore, Cineca in this SP is responsible for the data services activity, and the objective here is to focus on testing and deployment of tools and technologies for data management, and all this data management must be aligned with the workflow and the use case that we provide in human brain. Furthermore, we are involved also in a different activity in human brain. It's FEDAPA. FEDAPA is a federated data pilot project. You know that the big supercomputing activity, big simulation, is very related to the access of a huge quantity of data. So it's better to integrate near to the supercomputer also the possibility to offer a data storage, data activity at support of science. So the idea here is to federate a data activity to realize a pilot version of a federated data infrastructure at the support of the neuroscientists. Now this pilot is started between Cineca, CSC, as in Lugano and Jülich Supercomputing Center in order to federate our data infrastructure and to provide a common, a unique way to access the data. So perhaps we can, we have a cooperation with Lens to introduce data, to get data from Lens, to move them in Cineca, to access data, to use for simulation, and then perhaps to visualize those data from a different site, like in Aachen or in Madrid, and then provide other data that can be accessible from other scientists around the project. Here it was just an example of from Lens we can move through Unicore and Unicore FTP to the Cineca and access the FEDUP activity and work in cooperation with other centers. Furthermore, we are involved in different other activity. We work with scientists from the University Campus Biomedical in Rome with Professor Iannello in order to provide tool. They provided, we introduced the visualization and the parallelization of this tool in order to stitch the data that are coming from Lens in slides and peel up those data in order to provide a 3D raw data toward the side coming from the microscope images and stick, merge, and save those images in a multi-resolution representation, so from different slides to arrive to the three-dimensional, and then to try to analyze at different level of resolution in order to try to work on those images at the scale cell. We have provided also some converter for the data and some GUI graphical user interface in order to better access to this tool. Other cooperation come from the support to science in different simulation. Here there is an example of the olfactory bulb, and here there is another support to science in the field of hippocampus simulation. A talk by Michele Migliore later will explain this kind of simulation and so on. So, just to conclude, we are committed in a human brain project to provide a pillar in the field of the HPC and data analytics platform in order to support the best the human brain and the neuroscience activity around Europe. Thank you.
So I think we close this first session and we have a half an hour break. So we start again at 4:15. And for those that have not visited yet the science market, you can use this half an hour opportunity to visit. So let's see here at 4:15, half an hour. Thank you. Thank
problema di conversione.
Lo proviamo subito, ma almeno siamo a posto.
No, mettiti in alto, sempre peggio. Sì, poi
Devo tornare là. Sì, prego. Si vuole appoggiare. Il cavo arriva fino al bar. Possiamo metterci qui e io uso col mouse. Ok. Un attimo vado in regia.
Benissimo. Funziona, sì. Ah, deve girarlo lei?
Poi ci risentiamo perché c'era una persona con cui dovevamo parlare dei vostri per vedere. Vabbè. Fammi caricare la presentazione e vediamo che cosa passa.
visto Gabriella ieri.
I'm here. If you reach me, I'm here. No, no. I am inside of the room where. Thank you.
Welcome to the second part of HBP in Italy. We have a certain number of presentations. The first one is from Enrico Cherubini. Enrico, come. Exploring synaptic function with intrabodies against Gefrin and neuroligands.
Okay, good afternoon. So our project within the Human Brain Project is, our task is, has been already mentioned by Antonino Cattaneo, is the use of intrabody against Gefrin, Nerexin family to explore synaptic function and to develop a computational model of transsynaptic signaling. And according to a classical view, the information flow only unidirectional in the brain. The spike traveling along the axon arrive the terminal and depolarize the terminal. Calcium enter, there are exocytosis, transmitter release, and postsynaptic currents, and eventually a spike, so the information flow. But this is, in some way, should be revised, this, since we know that could be, is more complicated because there is a crosstalk between the post and presynaptic specialization. This could occur through two ways. One is classical second messenger, and the second is through adhesion molecule. Now, this is the example of second messenger. Classical second messenger is cannabinoids, which are released by the postsynaptic neuron, the pyramidal cells, and act on receptor present on interneurons containing CCK. And when act on this, they bind to the receptor and they suppress transmitter release. This is blocked by antagonists. Another way to block, this has been shown already by Antonino, is through adhesion molecule. Adhesion molecules are molecules of the neuroligin Nerexin family, which in some way interfere with PSD95 at Gefrin, which are scaffold protein at inhibitory excitatory synapses respectively, and they bind to the presynaptic partner Nerexin. And what they're doing very important because they are important for synapse formations or synapse stabilization, etc. And also some alteration in this molecule is associated with association in the gene encoding for this protein are present in neuropsychiatric disorder like schizophrenia, autism, etc. And there are several isoforms in human 5, in rodents 4, and mainly the more important one are the neuroligin 1, which is expressed mainly at excitatory synapses, neuroligin 2 expressed in inhibitory synapses, and neuroligin 3 about excitatory inhibitor. And in order to bind to neuroexin, they should dimerize, so they are dimers. Okay. Now, as Antonino already told you, we are using intrabodies. So intrabodies, the first that we have developed is an antibody against Gefrin. Gefrin is this scaffold protein which traps the receptor in the right place in front of the release site. And what we are doing, we are, so the antibody are developed mainly in Pisa, in scuola normale, and then we are clone this into lentovirus vector under the promoter, a classical promoter, and these are linked to, are fused to HFP, and also they are, in this case, they are retargeting into the nucleus of the cells. So they interfere with Gefrin, and once they interfere with Gefrin, they put Gefrin in the nucleus. So using culture, hippocampal culture, we have shown that culture with exposed to intrabody against Gefrin, you see that there is a reduction in number of Gefrin and GABA receptor cluster, mainly synaptic GABA receptor cluster. You can see here only EGFP, and this is EGFP with antibodies. And you can see that there is a significant decrease in the number and the size of the clusters of both Gefrin and gamma-2 receptors. And this effect is associated to a change in miniature events. We have a reduction in amplitude and also a reduction in frequency, and also there is a slowdown of the onset kinetic of the GABA receptor currents. These are just a quantification, and this is just a very simple scheme that has been introduced by John and Westbrook in 1995, in which you see that the gating property of the receptors, there are a part here in black and one in red. The part in black is the binding site, so one, the receptor, the agonist bind to the receptor, there are two molecules. This is the binding site is concentration dependent, and then there is a second part which is concentration dependent. So once two molecules bind to the receptor, they can either open the channel or desensitize. So the receptor moves from this part to this part for many, many times, and then eventually go down. What happened? Oh. Okay. So okay, thanks. Sorry, this. Okay. So we have explored what is the alteration in this slow down the rest time. So we have applied a very, very fast agonist application system through a piezoelectric, in which you have a complete exchange in less than 100 microseconds, and you can see that the response to GABA, a low concentration of GABA, does not change, but change the response to saturating concentration of GABA, meaning that affect only the second part, when after binding change conformation, and they can go through the desensitized or open states. And so in this part here, and we have demonstrated that this effect is due to a reduction in the desensitization, the fast entry of the sensitization state that is important for synaptic current. And since this time constant is equal to the reciprocal of beta plus d, that are this constant, okay, this means that in some way, although not completely intuitive, if you have less receptors that desensitize, are more available for binding again the agonist. Okay, in this case, they should be associated with an increase in amplitude of the receptor, and this is not the case because you have a reduction in amplitude. But this is due to the fact that you have two facts in the same time. One is alteration of the gating property, and the second is a reduction in innervation, in GABAergic innervation. Doing double patch experiment from a neuron with mixed synapses on another neuron which has the antibody against gefferin. You can see here that there are an increase in the ferritin rate, a reduction in the amplitude, a reduction in the probability of release. So you have two effects that you can see here. That you have one in one site. I mean, the gefferin intrabody just affect the gating property of the GABA receptor. Probably this is very interesting through the intracellular loop of the receptors. So there is a protein-protein interaction. So depending on the clustering, on the size of the cluster, this interaction could be more or less strong. This led to less desensitization of the receptors. And the second one is through the release, probably due to the fact that gefferin interact with neuroligin, and neuroligin with nerexin. Nerexin are involved are connected with protein responsible for GABA release. And finally, just to finish, is that we have with the group of Michele Migliore and Desiner in Palermo, we have modelized this, we have developed a procedure just to study this transsynaptic signaling inhibitory synapses, and this has been published recently, and there are two novelty. First is that use single events just to calculate the variability, intrinsic variability between synaptic events, and the second is using also this transsynaptic signaling that has not been used before. Okay, and this just to recognize, I mean, the EBRI, Scuola Normale, and Consiglio Nazionale di Ricerca in Palermo for their support. And thank you very much.
So thank you, Enrico. Now the second communication, the first from Francesco, And Francesco is from single neuron to whole brain or for functional optical measurements and platforms involvement.
Thank you. Come si va avanti? Si preme qui? Okay, good. So thanks, Egidio. So what I'm going to talk about is how important in the brain is to connect a single molecule. I mean, in this case, let me say single synapse or even single neuron behavior to the whole brain. And so I'd like to show you some approaches that we are following to make those kind of correlative microscopy, and maybe also the model we are working on. And finally, I will close with the example of a co-design project, which is exactly, let's say, horizontally span the HBP. So the ultimate goal of HBP, of course, is to bridge neuroscience, medicine, and future computing all together. And as we were saying before, this is a fantastic opportunity to have a transversal, let's say, kind of approach that is able to put together those kind of disciplines. And in the multi-mapping, multi-level, let's say, recording of the brain on large scale, let's say, areas on the whole brain of a mouse, lenses plays a role on this kind of multidimensional and let's say multi-scale measurements. It's a microscopic challenge because we must be able to detect very tiny details and then place those details in a larger environment, which is the dendritic arboring, for example, of a neuron, and this plays on a much bigger context, which is the brain, which is the same, let's say, things that saying that we need a huge optical dynamic range. That means that I'm able to make a photo of all of you, zoom in the only one which is sleeping, for example, and then zoom in each one of his cells, and then being able to have an enormous, let's say, capability in my sensors of the camera to zoom in until I'm able to reach every single cell of your body. This must be accomplished, of course, with the fact that we must generate multi-level maps. So we must put together fiber connection, neurotransmitters distribution, cell distributions, molecular maps, genetic expression, and last but not least, we must overlap those functions with structural maps because we want to connect structure with functionality. We at Lens, we are developing a technology as well, and so we develop a new customized light sheet and serial to photon microscope which have some technical, let's say, features which enables to have a factor of signal to noise larger of a factor 20 with respect to standard volumetric imaging technologies. And also we develop a new kind of clearing agents which are able to make the brain completely transparent. As you can see here, this is a mouse brain, and this mouse brain has become completely transparent. This is very useful when you want to make a tomographic, let's say, 3D imaging of the brain. This is an example of the first, let's say, reconstruction of the entire vasculature of the mouse brain. And this is the very new, let's say, results we obtained 1 week ago with one of those clearing methods which enables now to have a very, very high contrast, unreached so far in literature in terms of fluorescence. And this is a two-color image. In other words, we have a two-color that in the same sample, and the neurovascular coupling now can be really reconstructed at a single synapse level. Of course, it is possible to use this kind of tomography techniques also to trace other kind of cells. These cells, for example, are the activated cells. These are showing the early gene expression, and those cells are indicating the activity in the last 4 years of life of the mouse. So before being sacrificed, the mouse was doing a task, and this is a photo of the all brain, this is half a brain, all brain activation maps with a single cell resolution.
This can be applied to human sample, and this can enable a new kind of paradigm for a 3D digital histology, for example. So this is, let's say, something which can be very useful in clinics because, like tumors, this is a case of a patient from Mayer Institute affected by hemimegaencephalopathy, and the anatomopathology. They have a lot of information by looking at the 3D structures of the tumor, for example. But this is just to give you an example on how, for example, applying the same techniques on the human can be useful also for clinics. The nice thing is that you can rewash and relabel with different antibodies, so you can make a lot of colors. And this is an example in which we were all washing out some antibodies and relabeling astrocytes, for example. And then, of course, the correlation microscopy, it's useful when you compare with other techniques. This is a PLI, which has been developed by Katrin Amunds in Jülich, and we are doing correlation microscopy between 2-photon and PLI in order to help the reconstruction in 3D of the whole connection of the human brain. Of course, the brain has nice features that is putting together the morphology with functionality, and that's our final goal. And so functionality is very important. What we are doing, we are placing functionality in the frame of a pathology like the stroke. And here you can see here that we can induce in a very well-controlled way the stroke on the animal, and then at that point we can place the animal between a neurorobotic rehabilitation task. So here we can observe the electrical signal, for example, calcium imaging, and then we can record all those signals during the rehabilitation task. And if we are doing that, we want to study this kind of effect from the structural and morphological and functional point of view. From the most morphological point of view, we are studying the vasculature remodeling at the whole brain level, the structural plasticity in vivo, the functional plasticity in vivo by looking at the calcium imaging, and the interhemispheric functional remapping by using optogenetics. First results, very, very encouraging, is that if we are looking, for example, the synapse turnover and the surviving fraction, okay, we can see that after the rehabilitation, okay, we can restore the same surviving fraction and the same turnover ratio before the rehabilitation. So the rehabilitation is really changing this synaptogenesis. And very astonishing is looking also at the spatial orientation of the neurite. If you look at the peri-stroke regions, you see that before, let's say, the stroke, the orientation in the space is it's quite large. Nearby the stroke, the neurites are all oriented in one direction, and after the rehabilitation, you recover let's say, the sprouting. So it's very nice to see how really the rehabilitation is not only affecting spinogenesis, but is affecting also the orientation into the space of the dendrite. And if you look at the vasculature, it's even more astonishing. I mean, if you look at the vasculature orientation, here in false color are the 3D orientation into the space. Nearby the ischemic area, you see how the vasculature is oriented in one direction due to the ischemic damage. But then if you apply the rehab, you don't have the same recovery that you have in the neurite. So the neurovasculature coupling in this case must be well studied and simulated, of course, because in the Human Brain Project we are collecting those data to perform modeling and simulations in order to understand also those kind of simulation, how does it work in a pathology case. This is a typical signal from calcium imaging during the rehabilitation strokes, and you see how well synchronized are the neurons with the forces and with the position of the load cells. But then it's even more astonishing. If you study, for example, the propagation of the signals into the brain, this is the stroke area, and you make some isochrones. In other words, you see in time how those signal propagates, and you superimpose with the atlas in order that we have all the region of the brain, and then you make the cross correlations of the region of the brain.
you see that the stroke and in the rehab after 1 week, by indicating with red the very well correlated regions, you see that mostly regions are not correlated nearby the stroke area. Here is the bregma, and we are looking at those kind of regions. But when you have the rehab, you see that immediately you recover the synchronizations between the different area, like in the control. And so this is a very well, let's say, way of looking in a similar way with, as you do with the F-bolt signals with fMRI, but with, in that case, with a much higher resolution, at least superficial resolution. Then, of course, you can use optogenetics, and with optogenetics, you stimulate the left hemisphere, and you look the activity on the right hemisphere. So this is the contralateral functional connectivity, and it's nice to see that this functional connectivity, it's disturbed by the stroke, and it's recovered by the rehabilitation. So the rehabilitation works at the level of single spines, works at the level of orientation of the dendrite, but also works at the level of inter-hemisphere connections. So it's a real world that is connecting the 100 of nanometer with centimeters. So what we want to do here is we are coordinating a co-design project which is called the Development of All Mouse Brain Model. And the goal here is to develop a model for the mouse brain and to place that model also in an environment with a pathology like a stroke. So we will have 5 products which are products of the infrastructure. In fact, the co-design projects are going and are meant to realize the infrastructure of the Human Brain Project. And those 5 products will be the reference setups and the 3 applications. The 3 applications will simulate the anatomy, will simulate the imaging, and will simulate the behavior. And of course, they will apply to these use cases, but then can be generalized and applied by everyone as external users. Finally, we have a product on Data Explorer, and Importer. Those kind of products are intersecting all the platforms from the neuroinformatic, brain simulation, neuromorphic, neurorobotics, and high-performance computing platforms, and of course are intersecting all the different sub-projects of the Human Brain Projects. So I'd like to thank not only, the people that are in my lab working in this project, but all the collaborators that are spanning all over Italy, Europe, and the United States. And I would like also to thank you for your attention.
Thank you, Francesco, and also for being on time. And now we move to the next communication from Pier Stanislao Paolucci, simulation of cortical activity.
Yes, okay. The previous presentation provides a perfect motivation for the reason why an institute like the Italian National Institute of Nuclear Physics is starting to be really, really interested and excited about this initiative. In fact, previous presentation showed the fact that we are beginning to understand the structure of the brain at all architectural level. And doing fundamental physics basically has the aim to understand the law of the universe, and the frontier of physics today is complex system. So complex systems are those systems where out of elementary, let me say, particles, in this case neurons, synapses, complex behavior emerges. And all scientific revolution started when, for example, relativity, you start to look at the logic element, at the epistemologic element, and then at the mathematics. So the fact that you have information at all scales permits us to start asking questions about how the basics of mathematics is produced. For example, order relation, how the temporal arrow is created out of basic laws that are time symmetric, and plasticity with its temporal order relation starts to create a few glimpses in this direction. Okay, so that is the reason why the Italian National Institute of Nuclear Physics, which is the Italian agency for the study of the fundamental constituent of matter and the fundamental laws, started 6 years ago an investment in trying to contribute to this field, initially in the modeling area. Understand how we could simulate with high possible efficiency the brain as a system. Okay, among the assets of INFN, which employs all over Italy in all physics department about 5,000 researchers, in Roma we have a lab, which is the Hbp Parallel Computing Lab, which has been created in 1984 by Nicola Cabibbo and Giorgio Parisi, and I had the fortune to participate to this lab since the beginning, where we every time tried to challenge problems where high-scale numerical simulation are required, and the methodology is always the same. We try to capture the essentials of the numerical simulation on standard computers, then we identify what are the bottlenecks on a standard computer, and then we try to design new solutions, software and hardware, together, a co-design, to address this kind of problems. For example, already in 1992, if you look at the slides of Cineca before, it is a lot of time ago, we had already system in place with 2,000 processors running in parallel to solve theoretical physics problem. And we developed, you can see the picture in the bottom left, our own chips for accelerating computation. And actually, we put on the silicon the equations of the quark and the gluons. Okay, that is the idea. Understand the problem on a standard computer, then try to accelerate the simulation. So, furthermore, for us that are, let me say, designers of computers, the brain is a great challenge and inspiration because it is the ultimate computer. With 100 billion neurons and 10,000 synapses projected per neurons, basically you have an engine which is able to exchange, firing at 10 Hz or something like that per second. You have an engine which is able to send 10 to the 16 messages here and there. And if you would like to simulate that, you need at least several hundreds of thousands of computation per event. And surely more capability are yet to discover because at the synapse, for example, a lot more is happening. And the incredible stuff is the fact that the brain power consumption is only a few tenths of that. You can discover it easily. You put your hand on your head, and it will dissipate less than a conventional bulb. Okay? If you would like to simulate this stuff on a conventional parallel computer today, you will need, in real time, you will need several nuclear power plants and billions of processors. Okay? So, and power and parallelism are the challenging today computing platform everywhere, from your smartphone, where the application you can perform are limited essentially by the battery, to high-performance computing, where the limit is the cost of the power supply. So there are, in parallel computing specifically, a few topics that are really interesting. Understand those computational and architectural tricks that permits this efficiency. Design, in first instance, efficient software simulation techniques on standard computers, and then co-design brain-inspired hardware and software to accelerate this kind of computation. Obviously, one could ask a question, but if the problem is so tough, how can you aim to simulate the system? Obviously, we have to start scaling down the models. For example, simulating a mouse, which is much less complex than a human's order of magnitudes, or scale down area model of areas, or going down to a single area, or a scale down single area, or even to the single column. So, yes. So, to this purpose, we started a few years ago in cooperation with the National Institute of Health, ISS, a cooperation for developing an acceleration for a specific problem, which is slow waves, deep sleep in the cortex, which is a fundamental state that actually is incredible. If you take a young person or a young animal, the more the animal is learning, the more it goes in deep sleep. When you have an infant, it spends something like 16 hours per day sleeping. When you are old and you are unfortunately learning not so much, you spend a very few hours in deep sleep states. So when you are sleeping, you are not conscious, but it is fundamental to create consciousness. So, and the more that state is something very, in some sense, easy to start understanding and measure, because the behavior is more stereotyped and you can easily perturb it and observe what are the propagation of those perturbations in the system. So this is exactly the goal of the wave scale experiment that INFN is coordinating in cooperation with Roma with the Italian National Institute of Health, and with Milano with the team of Marcello Massimini, which is performing those wonderful experiments on assessment of consciousness. You have seen this in the previous slot. And then in Spain, we have experimentals on the mouse, so doing electrophysiology on the mouse and photostimulation and photoinhibition at very high resolution in the mouse. And here, what we plan to do is to start doing our parallel simulation on our own tool, but then we are cooperating with SP7, in particular with the team of Hans Eckerhard-Plesser and Abigail Morrison, to be able to port to the standard simulation environment of the Human Brain Project our simulation. So at the end of these years, everything we will have learned will not remain in a funny machine that has been developed on purpose, but will be saved on a platform that can be used by the community in the next years. So perhaps this slide is not so necessary, but because I already covered by Francesco Pavone before. But the idea is that we would like to understand the cortical organization, in this case of this sleep, starting from the lowest complexity mode. And for computer architect, the brain is an ultimate computer to challenge. And okay, there are some immediate technological impacts in Italy and in Europe, because by mastering what is happening, we are really capturing in our teams the knowledge that will be a key for the next year to come in a world where, for example, robots and artificial intelligence will play an increased role, not counting the 800 billion of euros per year of cost of the diseases that are brain-related. Okay, thank you.
Thank you again for this presentation. Now we move to the last two presentations before the coffee break. I would recommend to keep the time because we're a bit late. So, Maurizio Mattia, what can a sleeping brain tell us about its functions?
Thank you. So, I don't think that I need to convince you that sleep is important. But just in case you have some doubts, I can give you a couple of evidences. Part of them maybe are, you know. First of all, we spend a big part of our life sleeping. And as you see and as you know, the amount of sleep is decreasing with time. But in particular, what is interesting here is to see that the quality of sleep is changing. In particular, when we are newborn, in the early stage of our life, the two stages, the rapid eye movement sleep, has more or less, is a big component with respect to the deep sleep part, which is here depicted in green. Furthermore, this deep sleep is something, and not only the sleep in general, is something that has been conserved across evolution. Across 100 millions of years, sleep is there in reptiles, in birds, in mammals. And in particular, the non-REM sleep, this green, is very ancient. This is suggesting us something which is important. Sleep is a kind of, is needed, and is a kind of default mode which is shared among species. And so it is rather intriguing also to think about the fact that sleep is there and allows us to compare species. And let's have a look, for instance, to what's happening in a brain which is sleeping. This is a picture from a classic work by Mircea de Steride, in which you can see that sticking an electrode in the brain of a cat, you can see that at the microscopic level, electroencephalography show when the cat goes sleeping, alternation, the polarization of field potential. And at the single cell level, you see that when the cat goes sleeping, you have an alternation of the activity of the single cell. So it is not only at the macroscopic level, but also at the microscopic level, you have an alternation of periods in which the cell is not firing, and periods when the cell is firing a lot. These are spikes. Not only, these two recordings are from one hemisphere. These other two recordings are from the other hemisphere. This means that this brain, which oscillates when you are sleeping, is producing this oscillation in a very synchronized way. It's a whole phenomenon. And this is particularly apparent from this experiment performed in vivo with optical imaging in anesthetized rodents. You can see with this voltage-sensitized imaging that an anesthetized animal is capable to produce these rhythms in a rather coordinated way, like waves. Here, blue means low activity, actually here, and red means high activity. And during time, you see this planar wave or spiral wave, as in this case. So the same substrate, neuronal substrate, is capable to produce two different modes of alternation, so that cannot be distinguishable at the macroscopic level. But when you widen your point of view, you see that different things can occur. So seeing this kind of ways, the first thing that you can imagine is, okay, our brain, when we sleep, our brain is like a sea, a rough sea, let's say. There are waves, but this is not the case, because the sea with waves is there only because there is some energy provided from outside. There is the wind. Without the wind, you would have a flat sea. So the brain is something different when it's sleeping. The phenomenon is collective, but is complex, because the brain, when it's sleeping, is a kind of isolated system, and is capable to do this rhythm, this global phenomenon, endogenously, without energy provided from outside. And this is due to its complexity and due to its excitability. Complexity and excitability is the result of different levels that are interacting, moving from cell assembly, as in this case, that thanks to synaptic reverberation, as in previous talk has been shown, are capable to produce nonlinear amplification of the input coming from other areas, for instance. But in addition to this, you have columns at the mesoscopic scale. And this activity, which is going up and down, is spreading in a particular spatiotemporal pattern across the column. And taking together these columns, you will have waves which traveling across the cortical surface. And this is, as I told you, a complex phenomenon that has to require bridging the gap between all these levels. And this is where people like me, I'm a physicist, can play a role. Because looking at the results of the interaction between many simple systems requires to have a theory in mind, to have simulations. And this is what we do. In particular, we develop at different level of complexity models that are capable to produce this up and down oscillation, as the one I showed before from the stereo experiments, as in this case, these point are spikes. This is a simulation with 120 neurons. There are some neurons which are inhibitory, others which are excitatory, and then by themselves are capable to produce these oscillations. And this oscillation, when you put these modules together, interacting with some topological interaction, they are capable to produce the waves I showed before. In this case, these waves are due to the fact that there is an activation on a small piece of our cortex, then there is some fatigue, and this is shutting down. And actually, rather than a rough sea, the slow wave activity in the brain is more similar to a burning forest. Because when you have wood, there will be fire. But after the fatigue period, wood is gone, and then you have to recover and wait that the trees are coming back in order to produce the next wave. So this collective phenomena, as I told you, requires some theories, the capability to see how elements put together are capable to produce unexpected phenomena. And, but of course, the gold standard is to have experiments and theory together. And this is why in the project highlighted by Pier Stanislao Paolucci, we have a tight collaboration between experimentalists and theoreticians like me. In particular, this is results in our lab in collaboration with the laboratory of Maria Victoria Sanchez-Vives in Barcelona, in which we see how these waves can tell us something more. Not only the fact that, okay, the sleeping brain is oscillating, but also that these slow waves are shaped by the structure. So having a look to the slow waves maybe can tell you something about the organization of the brain. And this is exactly the case. We have, as a paradigmatic experiment, we have a slow wave activity in a cortical slice. And we reproduce the large majority of the features of this spontaneous activity in a model, like in this case, you see the spreading of the activity. And we see that if you have a strip of excitable cells, which is here depicted by this dashed line, you will have waves that are moving in opposite direction, but follows always the same pathway. And this same pathway, we expect to be close to the strip where you have the more excitable part of the slice, corresponds exactly in experiment to the place where you can find layer 5 and layer 4, which is known from the literature that it is the most excitable layer in the cortex. This is only to tell you that this kind of default mode, the slow wave activity, which is in any species of animal, is a very good reference, experimental reference, because can give you the possibility to see how the different levels of description, the different dynamical levels are interacting, and which are the features of the different levels that, when they are interacting, are capable to produce this complex phenomena. And of course, the next step, once we have characterized the multiscale organization of the brain of a sleeping brain, the goal might be to see how the same substrate is capable to produce computation, to perform sensory processing. But this is another story. We are working on that, and many people are working on that. And only to tell you that this is why sleep is so important, because maybe it's a default mode, simple mode, but complex enough to crack the organization of this nice system, which is our brain. Thank you.
Now, Michele, the last talk, the brain simulation platform, how it can be used to advance our knowledge of the brain.
Okay, thank you. I'm promised to keep it short, so I don't want to interfere with the art performance later this evening. The brain simulation platform is that part of the infrastructure that allows a user, internal or external to the Human Brain Project, with the right proper accreditation, to run in silico experiments using large brain regions at different scales. In particular, I'm going to talk about cellular level regions, models. My lab is involved in different areas of the Human Brain Project. As Enrico Cherubini told you before, we are also involved in the transsynaptic signaling, also some technical inner working of the brain simulation platform, and also in the hippocampus and in one of the co-design projects. So the main goal of the brain simulation platform is to develop applications and web services to support collaboration, basically, to build, simulate, analyze, and validate, and disseminate data-driven brain model. These are 3 examples interacting with different sub-projects in the human brain project, the neocortical, cerebellum, as you have seen before from Egidio, and also the hippocampus. There are several occasions in which you can see live demos, hands-on activities, and more on Friday during the deep dive sessions, also in the parallel sessions. So the nice thing about the implementation of different brain regions is that within the brain simulation platform, we use more or less exactly the same circuit workflow. So this would allow a bottom-up approach, which is kind of aseptic with respect to the specific model that we want to model, that we want to implement. Of course, there's different blocks. I'm not going through all of them, of course. The different blocks might be dependent on the brain regions that you want to model, but the kind of activity that is implemented to start from the data and arriving at the circuit is the same, independent of the circuit. And so, since I'm going to talk, I'm more involved in the hippocampus. Just let me give you more information for the general audience that it may be not informed or aware of what the hippocampus is. It's a brain region. One of the oldest brain regions, deep inside the brain. It's composed by a few million neurons. It is strongly involved in the higher brain functions like learning, memory, spatial navigation. And most of the major brain diseases starts from there. Alzheimer, epilepsy, and also cognitive aging, and all a bunch of other very important and severe diseases. What we do as an example for the hippocampus is to implement a kind of integration of different platforms. For example, for the hippocampus, but also for the cortex, and what we're doing also with Egidio for the cerebellum, is to use data from SP1 or 2 that are in the neuroinformatic platform, in SP5 then, and then use tools in the SP6 to build the circuit, and doing the in silico experiment in the simulation are done in one of the supercomputer centers, and then use the results or the model itself in other platforms to reduce it and eventually connect it to a robot for a variety of reasons. Now, if everything goes well, I should be able to show you an interactive visualization of a simulation that was built with tools developed within the platform in SP6. The code was deployed and run on Jülich supercomputers, and now we are looking at the simulation by using the remote connection, the visualization from Cineca. Probably the resolution is not good because the resolution of the projector is low, but we'll see what happens. Okay. So now we are connected, and we run a script, and we should have the simulation, the visualization of the simulation of the hippocampus. So one part of the hippocampus, actually, the CA1 region. There you go. So these are about 1,000 cells, and the entire hippocampus, the entire CA1 or CA1 regions of the rat is about 350,000 cells. But and this is interactive. I can move it with a mouse, doing zooming in and out. And this would allow a user to enter the platform, configure the simulation, choose the region, choose the kind of input, choose the plasticity rule, the synaptic integration, everything, run the simulation and visualize it after it is run. And this would allow some kind of actually virtual experiments on very realistic cells, very realistic morphologies and synaptic connectivity. You can study all sort of self-organization of this network. For more specific, better visualization, we'll see for specific region, we can also produce movies. Oh, sorry. Maybe you have already seen the movie downstairs during at the booth, at the SP6 booth. Let me see. Yeah. So in this case, this is not interactive. This is a movie, but we can build it at much higher resolution and with specific directors' suggestions in such a way to visualize one region or another region, some specific neurons or kind of activity. And other thing, not especially presentation, of course. Okay, of course, this is not done in my lab alone. Of course, there are several people, especially at the EPFL, a large group of people that I'm just listing here a few. And I would like to especially thanks Cineca for the kind support in all parts of this kind of, both in the simulation and in the visualization, and that has given the platform so far. Thank you.
So thank you, Michele. I pass now. I pass now to Francesco for the end of this session.
We have some debugging problem.
Yeah. Computer problem.
Unplugging problem. So let me make some closing remarks, and let me say that I mean, for me it's great to have these distinguished colleagues and friends here talking of these things, and I mean I'm impressed by the fact that I mean how strong is the effort at the international level. But let me say also with some national pride, I'm also impressed by my colleagues and friends of HBP Italy, and I think it's a great opportunity for Italy, but it's also a great opportunity for all of us at the European level to work all together. So I'm extremely proud to have the honors to host this, all you guys here, and to host the first Open Day here in Florence. As you've seen, the brain is beautiful. The brain is beautiful also from let's say, an imaging point of view. So we have seen those images, we have seen those presentations, and at the science market today as well. Tonight you will see another beautiful visualization merged with music. We are thrilled to welcome the artist Nathan Sinigaglia, who is a visual artist. In this performance, Ravel Landscapes fuses in generative images and classical piano. By application of a custom software, the polyphonic sounds of the piano is analyzed and transformed into complex visual compositions in real time, similar to what HBP is working to achieve with the simulation of the human brain. So we will have now a brief, let's say, break, but at half past, I think it's 7:30, yes, 5:30, sorry. So in 10 minutes, you have to go to see this art performance to auditorium, which I think it's in the other buildings. And I hope you can enjoy it. So thank you for coming and goodbye, everyone.
Diciamocelo pure, sì. Io quello veramente d'accordo.